Our first speaker would be Dr. Karishma Kaushik. So let me tell few words about her. Dr. Karishma Kaushik is a physician scientist who received her MBBS from Maharashtra University of Health Sciences, MD in clinical microbiology from Armed Forces Medical College, Pune. She then earned a PhD in molecular genetics and microbiology at the University of Texas at Austin, where subsequently she served as an assistant professor of instruction in biology for some time. She returned to India as a Ramalingaswamy Fellow in 2018 and established her research group at the University of Pune. Her laboratory studies complex infection states with a focus on wound infections and biofilms under human relevant conditions. She uses a wound infection on a chip platform to get insights into complex microenvironments that influence wound healing and infection. Her talk today will focus on wound infection on a chip to dissect host-pathogen interactions and evaluate novel therapeutics. Let us welcome Dr. Karishma Kaushik.
And welcome to all of you. Great to see you here. Good morning. After an action-packed and very busy first day, we are embarking on the second day of the program. And I have to say, it's my talk that stands between you and coffee break. So the more questions, I think the faster we will get done. All right, but I hope it's exciting and entertaining, and I hope you really get a glimpse of the kind of work we do. So I'm Karishma. I'm a physician scientist based at Savitribai Phule, Pune University. I returned to India in 2018 to establish my independent research group there in 2019. And since then, we've been involved in a range of research endeavors as well as larger contributions to the science ecosystem in India. And I encourage you to take a look at our website to have a glimpse of all that we are up to. And it's pretty updated and we're very active. I must also thank the organizers and the funders for providing me with a childcare grant to be able to be here with my son. It's Diwali school break. And this was a much needed provision because truly thinking about it with him at home, it would have been difficult for me to be out for a whole week. So thank you very much, and I hope we see more of this in the future. All right, so all the work I'm going to be talking about has been done by our team. The wound infection on a chip research focus has been led by Nizam, who is there somewhere in the audience, right? And you can meet him after the talk as well. A lot of the advanced image analysis has been done by Shriya, who is also there in the audience, and you can catch up with her. But I must say that this has been a 3.5-year-long project, and there have been many different parts, which I'm going to talk to you about in building this integrated wound infection on a chip. And so there have been many people who've worked on it over the years, and it's been really Nizam who's held the whole project together. So we are funded by DBT, and I will also talk a little bit about the funding at the end. All right, so are we ready to deep dive into the platform? Okay, so yesterday we talked about organ on chips, a lot about organoids, more in the context of non-infectious states. So let's take a few seconds to think about infections. Similar to many disease states that are not associated directly with pathogens, infection states are actually very complex microenvironments. So it's not really just a bug causing pus, it's much more than that. And if you think of either the lung infection, the gut infections like colitis, or wound infections that I'm going to talk about in more detail, they're actually very complex, very dynamic, and they have multiple players that are involved that influence the progression and outcome of the infection state. So some of the key players that are very intuitive would definitely be the pathogen. I'm going to introduce some terms like multi-species pathogens and biofilms to you shortly. We also obviously have host cells. They produce matrix factors like collagen, fibronectin, fibrinogen. Then you have metabolites like lactic acid, lactoferrin, short-chain fatty acids, which are all bathing the infection milieu. And a less addressed, and I hope our work is able to address that, is the presence of biophysical factors. So we don't really think of infection microenvironments to be dynamic in that sense of biophysical factors at play, but actually almost all infection sites will be bathed by fluid milieu, which will exert shear stress, fluid flow, and wound infections sites are also subject to compression and stretching forces, right? So we are going to look at whether we can recapitulate key biophysical forces in our wound infection on a chip. So I think I've taken you quite a bit into what I'm going to talk about. But let's think of this, right? Chronic wounds are actually a global problem with rising diabetes among South Asians, obesity, increased life expectancy, and hypertension. We have a lot of patients presenting to the clinic with wounds that don't heal. So a chronic wound is anything that doesn't heal in over 3 weeks of time. And this is a huge morbidity as well as a cost to the healthcare system when they keep coming for changing their dressings, debridement, and new antibiotic approaches. So typically, we all know this. This is cell biology 101. Wounds are supposed to heal in a very well orchestrated, coordinated fashion where you start with a clot and end with a scar, and that should happen within 3 weeks. And chronic wounds are arrested in this phase of inflammation and proliferation where they fail to progress through this orchestrated, coordinated series of events. And one of the most important factors that results in this stalled progression or this arrest in the inflammatory to proliferative phase is an entity, a microbial entity known as biofilms. So biofilms are these, they can be described as microbial consortia or microbes living in cities, So they are dense mats or aggregates of microbes. They're not free-living single-cell microbes that we think of when we grow them in a test tube under shaking conditions in the lab. But they're actually very dense mats or aggregates and that are found in the wound infection site. So if you look at clinical biopsies of wounds, this is how aggregates will look. So the blue is all the host cell nuclei, which are your fibroblasts and keratinocytes. And the red and green are fluorescently labeled bacteria, which is seen as these aggregates or clumps, what we call biofilms. It so happens that Pseudomonas and Staphylococcus aureus are the two most common wound pathogens, wound biofilm forming pathogens, and they're often found together. So not only is the arrested wound an infection state, but it's also a mixed species infection state. So you can see where we are going with all this build-up. So in the chronic wound microenvironment, what do biofilms, where do biofilms live, right? What does the whole ecosystem look like? And if you see, you can think of it as, there's this wound bed, and the wound bed basically consists of human cells, host cells, fibroblasts and keratinocytes producing matrix factors like collagen, fibrinogen, fibronectin, and it interfaces with the immune compartment lined by endothelial cells, and this will basically be neutrophils. Could also be monocytes and macrophages at different points in time. But what you have is aggregates or biofilms growing on this wound bed, host cells and matrix, and then you have biophysical factors such as wound fluid, which is flowing over this wound bed. I also mentioned stretching and compression forces. So one thing that we are going to want to recapitulate in our wound infection on chip is wound fluid and the presence of flow or shear stress that wound fluid will apply when it is flowing on the wound bed. All right, okay. So why do we talk about wounds and biofilms so much? Because biofilms are notoriously resistant to antibiotic treatments, and we basically treat them by throwing everything at the biofilm, and we just hope that there is some positive outcome, and if not, we try new drugs and surgical debridement and dressings. So biofilms, in contrast to single-cell bacteria, are extremely resistant or tolerant, rather, to antibiotic treatments. And so we are constantly trying to find combination approaches or novel antimicrobial approaches to help eradicate or inhibit or kill the biofilm. All right, so there's a lot of background and a lot of reason to study wound biofilms, but where does the field of wound biofilm model systems stand? So if you think of all model systems as in vivo, ex vivo, and in vitro, in vivo, we often have rat and mouse models of wound infections, but not only are they technically and scientifically challenging simply because mice wounds do not heal like human wounds. And this is not because humans are not 70-kilo mice, but because actually mice lack a layer in their skin, which means that their wounds heal by contraction and not proliferation. So really it is not a scientifically accurate model. There are also ethical and cost and availability issues with being able to infect animals in an animal house, which is challenging. So to overcome that, we have a lot of ex vivo porcine or human skin models. This is usually from liposuctions or abdominoplasty kind of surgeries that discarded human skin. And they are rather technically challenging, though they offer the ability to be able to actually burn them or wound them and then keep them in the lab for prolonged periods of time under optimum nutrient conditions. But honestly, acquiring these, in India, we don't have abattoirs or offal houses where you would get quality porcine skin that is fits laboratory protocols. And so there's a large amount of work being done in building in vitro models like wound fluid models, wells, reactors, microfluidics. Okay, so there's a lot of work being done here. And we are moving towards developing a macrofluidic wound infection on a chip that's an in vitro model. So when it comes to in vitro models, people start with building wound milieus or wound fluids, and then they take it into some platform which can hopefully recapitulate more features of the wound infection microenvironment, like host cells, matrix elements, biophysical factors. So there's definitely limitation to having reductionist in vitro models, and we need integrated or composite in vitro models to be able to recapitulate more host relevant or human relevant factors, and that's where our work is going. So this is the summary of everything I'm going to talk about subsequently. So we have built a macrofluidic wound infection device. It looks like this. We can grow host cells. We can grow biofilms on the host cells. The host cells so far are in the form of scaffolds or fixed cells, but we would like to eventually move towards live cells. Interestingly, we can flow a wound milieu, which we have also built in our lab, through this device. And we recapitulate not only the nutrient and biochemical profile of the wound infection state, but also the biophysical forces. And at the other end, we can pull out all these relevant features, some of which I'll talk about how we've pulled out. Importantly, this entire setup is the size of an incubator. It can fit in an incubator. And this platform can be taken and imaged under a confocal microscope. Right? And you can image it, take your readings, put it back into the incubator, and go on and so forth. Okay, so let's get into our platform. So all of this work has really been pulled together by Nizam, so he deserves the center place in this slide. Nizam is also looking for PhD positions, so you can get in touch with him. He is among you. All right, so let's think big, because when we want to recapitulate a wound bed, we want a vibrant or a robust growth of cells. So what we've done is using 3D printing, we have built this macrofluidic device. It has a central wound bed area where we grow the host cells, fibroblasts and keratinocytes. It has an inlet, an outlet, and it's going to be hooked onto a pump. Importantly, it's only 1,000 rupees to 3D print this device. They are pretty reusable a few times with ethanol wash and UV sterilization. Nizam can elaborate more, and the whole thing can be mounted under a confocal. So this is our macrofluidic device. We have built these dimensions of the wound bed based on clinical reports of average wound dimensions. And they obviously vary depending on the site of the wound and the duration of the wound, but we've looked at some numbers while building them. Importantly, the dimensions of this device allow us to recapitulate shear stress at parameters comparable to that reported in wounds. And we'll talk about that in the next slide. But before that, somewhere along the 3.5 years, I remember posting on Twitter that, peristaltic pumps are really expensive in India. And any engineering students interested in trying and building one with us? And then we had some students at our parent institute, Institute of Bioinformatics and Biotechnology, saying, all right, we can try. So what we've done is built an in-house peristaltic pump. And I think this is really important to draw a point here that while we talk of organ-on-chip and organoid systems, this is largely driven by the global north. And in the global south and in LMIC states, we do have cost concerns and cost crunches. So I'm really interested in seeing how the global south can overcome these challenges. And if anyone is interested in writing a white paper or a policy paper, I'm happy to partner with you to understand the challenges and how we can overcome them of doing microphysiological system work in the global south. Anyway, we built an in-house peristaltic pump consisting of an electronic controller, a motor rotor. It's fitted to a power bank, it's fitted to a battery. This is your car battery. So we went and bought Exide batteries, quite literally. So the whole system is hooked up. The electronic pump can be controlled from a laptop, and it has different modes, pulse, purge, and continuous, which flows through the pump, the tubing, into the device, out of the device, and we collect the spent media. While it can be battery or electronically operated, there are challenges with hooking up something to the electrical supply when it's an incubator. And so we usually use the battery mode, and that usually runs for about 8 hours at a stretch, but we do have a backup power bank, and we have another battery replacement. Now, when we built this, it's much less than what a peristaltic pump would cost in India, which is anywhere between 1 to 1.5 lakhs. But we also said, maybe we can publish this as a standalone paper, where we say we built an in-house peristaltic pump that's 12 to 15,000 rupees. And so we did a lot of calibration on our system, and with several tweaks that were necessary. And when we publish this, which is the next piece of paper I want to dive into, we're going to be full disclosure about what were the tweaks and changes we needed to make at different points in time to make this pump work. But now we've achieved substantial calibration from 50 microliters to 500 microliters in battery-operated continuous mode. Now, what's very interesting is we can achieve flow rates from 30 microliter per minute to 2 ml per minute. And when we convert this to shear stress using the, certain features of the wound milieu that I'll talk about shortly, we are able to achieve a shear stress in dynes per centimeter square, which is pretty close to what is reported in a rabbit ear wound infection model. So this is quite encouraging. And I have to say, we haven't measured shear stress directly. We talked about this yesterday. We haven't measured it directly in the device, but we have estimated it based on the dimensions of the wound bed and the properties of the fluid that we are going to flow through the wound bed. We've accounted for the thickness of the host cells as well. All right, so we're not very off the margin, but I won't say this is a very accurate estimation either. I have to be honest about that. Okay, so what we could do with our device now, if we say it's biomimetic, which means it has to support biological elements, so it's able to grow the two cell types relevant to the wound. And we also published this little side story that happens, when you're taking on a large project. And we found that we could reduce the content of serum and develop a composite medium with reduced serum. So it could come down from 10% FBS to less than 2% FBS in some cases. And we could co-culture the two host cell types in proximity with each other, which is actually very challenging. Because though fibroblasts and keratinocytes exist in close proximity in the skin, it's actually difficult to grow them in the lab together. And we were able to do this, and we published this as a side paper. But apart from that, what our device is able to do is support the two cells in co-culture. We make a scratch, and the scratch closes. So the cells are also proliferating and migrating. Is that my alarm? Okay. That would have needed to rush at that point. Okay, so as we move forward from the device, I remember talking to Snehal, Vandana, and later Nizam that, all right, it's great, we have a device, it's recapitulating shear stress, we've cut costs on a pump seems to be calibrating well, but what are we going to flow through this device? I mean, if you're going to finally flow MEM, then how human relevant is this? And our whole pitch is that it has to be human relevant. So then, with Snehal and Vandana, we looked at clinical case reports from nursing studies, and we found the profile, the nutrient and biochemical profile of wound fluid. So this was just nursing reports that had measured lactate levels or lactoferrin, etc. And then we said, can we make a similar milieu in the lab? So this is again, if you think of preclinical models as ultimately you have this integrated body on a chip, where liver, lung, heart, brain is all connected, but at the other end you can have reducing serum as an alternative to animal methods, which is human relevant. You can also have in vitro milieus, which recapitulate biochemical and nutrient profiles, remaining more human relevant than laboratory media. And so we work in different stages of this preclinical pipeline to eventually reduce, replace, refine animal models and animal use. So we said, all right, we came up with a composition which said that with 70% FBS, we would cover most of the components. What we need to add are these factors, these matrix factors, these biochemical factors. We measured the pH, specific gravity, it lined up well, and then we did our subsequent work in comparison with laboratory medium to say, well, we are showing different results than laboratory medium, as well as with just FBS, because FBS was the base component. So what does it tell you about the role of these other factors? That was an idea that we had when we added the FBS only sort of control. Okay, so we've also published this work, but to tell you very briefly, we grew biofilms in the in vitro wound milieu. The biofilms under mixed species, Pseudomonas and staph, showed robust metabolic activity in the in vitro wound milieu, which is the black bar. When we measured viability using colony-forming assays, we could see Pseudomonas grew well. Staph also grew, but you notice staph is less than Pseudomonas. So this was an interesting finding because it's well known that in wounds, Pseudomonas outcompetes staph. And so we could actually recapitulate this co-species interaction between the two pathogens. Okay, so we measured viability and metabolic activity, but what about antibiotic tolerance? We could also find that the wound milieu showed increased antibiotic tolerance compared with laboratory medium. Yes, in the case of Pseudomonas, it may be serum plays a dominant role in this antibiotic tolerance. And that apart, we could see that the wound milieu produced or led to the formation of antibiotic tolerant biofilms. Okay, so how did the biofilms look? When you grew them under single species, you can see staph and Pseudomonas are both forming nice mats. This is in your 96-well black plate under the confocal microscope, so you get good mats. But what happens when they grow under mixed species conditions? Under mixed species, you see Pseudomonas is clearly outcompeting staph. You do see staph, but you see them as these small aggregates pretty close when you look very closely. And this aligns with what's well known about the interspecies interactions between these two pathogens. So I think at this stage I felt we had enough to push this out as an in vitro wound milieu paper, and it's received quite a bit of attention since then, and it's also led to our third piece of work in the project. So one thing that I truly believe is, we publish when the story is ready to be told to the community. There is no waiting for high impact and fancy papers. If it's a story that needs to be told, we should tell it. All right, so now we have a device and we have a wound milieu that's supporting biofilms under conditions that mimic the clinical infection state, at least with regard to interspecies interactions, metabolic activity, and antibiotic tolerance. Then Radhika came into our lab as a research fellow, and I said, all right, the wound infection on chip will get made, but we can do something more in the meantime. I said, what if we take host cells, fixed albeit, a scaffold, what if we take our milieu and now see if we have a more tractable, relevant platform to probe a fine-tuned analysis of the structure and organization of biofilms, which we absolutely cannot do under clinical biopsy conditions. And that's what we call the 4D microenvironment. So they're basically host cells, which we've co-cultured with our reduced serum methods that we published in 2020. We've added a wound milieu, which we published in 2021. And together, we refer to it as a 4D microenvironment. The fourth dimension, so the 3D is the three-dimensional host cell scaffold. And we've done some analysis. That's all Shreya's work with a very advanced tool, Biofilm Q, where we've shown that they're three-dimensional in terms of their size, shape, arrangement. The fourth dimension is the in vitro wound milieu. And this sort of aligns with your 2D cell culture, 2.5D cell culture, 3D cell culture, and 4D is when you add a biophysical factor or a biochemical factor. And here our fourth dimension is the wound milieu. Okay, we said we have this, it's a 96 well, it's a static system, we are getting to the dynamic wound infection system, but can we now just grow mixed species biofilms? Let them be fixed scaffolds, that's fine, in the presence of the wound milieu, and can we probe biofilm structure and architecture? Okay, so we found some very interesting things, and this is all advanced confocal work which we did at NCL Venture Center. So I must talk about what the role of incubation parks do in fostering academic research. It's truly two ways. It's been excellent technical support provided there. So what did we find? We find that Pseudomonas grows to form these dense mats over time, which is great. We find that staph, however, retains its aggregate structure over time. That's interesting, because staph alone was growing these mats, right? We looked at that, and we've shown this data in the 4D environment as well. It's not on this slide. But Pseudomonas forms mats, and staph retains its aggregate structure. So we said, all right, we can now compare this with only the wound milieu, and we also have a comparison with LB medium, which is in the published paper. And we can now see what are the differences that this 4D state with host cell and with wound milieu makes to this biofilm structure and organization. And I must say that this is really impossible to do in a clinical biopsy. Right, this kind of fine-tuned analysis. So we got all the imaging done, and then we really needed somebody who could crack, if that's the right word for it, who could basically make tools, image analysis softwares work. And then Shreya was already with us on another project, which you can look up on our website. But I said, Shreya, I have a project for you. I think right now our research fellows are very worried when I contact them, because it's always some new project or some new idea. Okay, and so we worked with two tools, ImageJ and Biofilm Q. And we said, all right, Shreya, these are all the images. What can you do with this? The first thing we wanted to see is what is the size of the aggregates. And you see in the 4D microenvironment, Pseudomonas forms larger aggregates compared with Staph aureus, right? You see Staph aureus forming a larger number of smaller aggregates. So you can see this is a very fine-tuned analysis we were able to do with ImageJ and Biofilm Q, because we're really getting aggregate sizes in micrometers square. And we could say that Pseudomonas was tending to form larger aggregates early on. Does that possibly give an advantage to it growing into these huge mats? We don't know. We also found that when you compare with wound milieu only, the distribution of sizes of Staph aureus aggregates is influenced by the presence of a 3D host cell scaffold, because in the absence of the scaffold, the sizes are different. Presence of the scaffold, you see this shift to larger sizes. So could the presence of the scaffold, albeit fixed, but it is still a biomimetic substratum, it's not polystyrene. So could the presence of the scaffold be giving Staph aureus an opportunity to attach and grow, and therefore it grows into these larger aggregates early on? That's possibly something we could explore. Then we said, what about, so we've looked at one feature, aggregates, individual aggregate sizes. What about the location of the two species with respect to each other? And we could, as expected, find Staph aureus overlaps with Pseudomonas much more than Pseudomonas overlaps with Staph. This also makes sense because Pseudomonas was growing into these larger aggregates and mats, and Staph aureus was staying as smaller aggregates, so there will be many regions of Pseudomonas not in contact with Staph. All right, so we say they are in close proximity, the overlap fractions do vary. Now what about vertical spatial organization? And all of this you can pull out from a 96 well with your image analysis. So what we find is as expected. Early on, they are well mixed, Pseudomonas and staph. But over time, you see staph getting retained or embedded in the lower regions of the biomass, and Pseudomonas was growing to form these huge dense mats. And we see that similar in the in vitro wound milieu only, which possibly underscores the role of the milieu rather than the host cell scaffold. So we were able to pull out this spatial organization. And finally, when we look at biomass composition, so now we've moved from different levels of organization, from aggregate size to co-localization to spatial organization, and now the whole biomass and interspecies interactions. And when you look at that, you can sort of see that Pseudomonas is outcompeting staph, which you see here in the wound milieu only. This is the ratio of Pseudomonas to staph. Pseudomonas is outcompeting staph in both conditions, and you also see that Staph aureus under mixed species conditions seems to be inhibited over time, whereas Pseudomonas, the red bar, sort of remains the same over time, but Staph aureus is inhibited over time, which again points to Pseudomonas inhibiting staph. But when we see this in LB, which is in our paper, you see the Staph aureus drastically drops. So in LB, Pseudomonas rapidly kills almost the entire population of Staph aureus. So when you have a human relevant microenvironment, you're actually able to support stable coexistence and yet dissect interspecies interactions that you see in clinical wounds. Okay, so again we had a story to tell and we decided to tell the story. What we found, and I presented this at Biofilm Spain, and these were the people in the audience, so it was quite a moment for a young group. But what we could do with this study, with this data, with this information, is actually bridge the current model of wound biofilm organization with respect to these two pathogens. So the current model says that wounds are initially colonized, either randomly with both pathogens or one pathogen after the other very quickly. The current model immediately shifts to the non-healing prolonged state where you see this non-random distribution or this spatial segregation where Pseudomonas migrates. It's a motile bacterium. It migrates to the deeper regions of the wound and Staph aureus remains as aggregates on top of the wound. But obviously, this final state is a result of many factors like tissue remodeling, immune factors, inherent properties of the bacterium itself like Pseudomonas. And so we said, what we think we can do is after this initial coexistence, what we think is happening is, so they're initially coexisting, Pseudomonas is forming large mats, Staph is forming aggregates which migrate to the lower regions of the mixed species biomass. And what happens eventually, there is so much Pseudomonas biomass that it produces elastases that are able to break down the wound bed, and Pseudomonas migrates into the lower regions, this dense mass, and you see large aggregates of Pseudomonas in the lower regions and small aggregates of staph in the upper regions. I will say we haven't tested for elastase production in this system, but it's well known that Pseudomonas produces elastases under clinical conditions. So what we were able to do is sort of bridge these two well-known pillars of biomass organization in the wound bed with our work. All right, so we have many different components together, and what Nizam is right now working on is, in the device, we've grown host cells, we fix them, we stain them, and now we add our mixed species biomass and we add the in vitro wound milieu. So since this is a fixed scaffold, we are able to keep the biomass alive for a longer period of time. Professor Ingwer spoke about the challenges of live host cell microbe co-culture. But what we've been able to do is, it starts off like this, both of them together, and then you eventually see Pseudomonas out-competing staph, and all of these images will now go to Shreya, and she will apply the well-known tools we've built with ImageJ and Biofilm Q to parse out the features we've talked about and possibly even get new features and new insights, because what this system will allow us now is the presence of shear stress. So integrating all of this together, our device will grow host cells, fixed scaffolds, microbes, co-species. We will flow in vitro wound milieu under shear stress conditions with the peristaltic pump we've developed. We'll pull out readings like biofilm organization and biomass, structural functional readouts to say, is there decrease in biomass, change in biomass structure, could that be contributing to evaluation of anti-biofilm agents? And together we think it has a place in the preclinical pipeline for wound biofilm studies. And definitely we have several alternatives to animals that we've developed in the course of this work. So we are at an exciting point in this project, which we are going to take forward. But we've also had many small wins and small publications that come out of it, and that's been really heartening for our young group. I must say, as I end, that all this work has been done by research fellows in our group. They are not PhD students, they are research fellows, undergraduates, and master's students. We've been a very open lab. We try to create an opportunity for whoever emails us, and in doing so, we've been able to turn it into a mutually beneficial experience. So hats off to doing science with young people. Thank you.
Thank you. Not only is a nice talk, you perfectly finished in time actually. I didn't
That sounds very good. I worked on it.
Yeah. Now it's open for questions. Let us start with Dr. Abhijit.
Maybe I'm wrong, but the general assumption is a wound has an air interface. It's an air-liquid interface.
Correct.
But your model is not mimicking that.
Yeah.
Don't you think?
It's true, and I probably will need transwells to mimic that, the air liquid
So I
interface,
think
because
you
you're
should
able
be
to
working with, so when you talk about shear stress, it's very different when you have an air-liquid interface versus a liquid-liquid interface.
True. It is very true.
So I think probably you'll have to think
about that. Yes, and with microfluidic
or
macrofluidics, that is a concern. How do you create an air liquid interface even in a too well model?
Exactly. But see, the final question you're addressing is of open wounds probably.
Yes. But
Isn't
also
it?
there is an upper layer that is exposed to the air, but there are several deeper layers where in fact even anaerobes grow.
Yes.
So they are far away from the air liquid interface.
That's true.
Yes.
But
yes. We can think about
actually
it.
have, if you actually have a total liquid shear flow through the whole system, I think it won't be the best.
Yeah, we can think about modifying the device a little bit.
That,
okay.
Hi, Karishma, here. Yeah, fantastic talk, as usual. So just to start from, what Radha said, so the air-liquid interface, Deepa is here. So she has made a air-liquid interface for the skin model. So you can talk to her and then probably, you can take that one. So, but now I'm coming to my questions.
Sure.
So first one thing is that the shear stress that you talked about, that the rabbit ear. So how people measure that in vivo condition? So that is my first question. Maybe I can just complete my question and my second question is that in the physiological system, does this both the species of pathogen comes at the same time or one come first and the other come next? So do you need to model that in your system? And my third thing is that for your shear stress, because you are using a rectangular thing, the equation that you are use that will give you a rough idea but not a very specific answer. So if you are interested to do a computational fluid dynamics model, I think Sheetal is there and she can help you also. Yeah.
Fantastic, Abhijit. Thanks for all the points. And our expertise is not computational simulations of shear stress, but we can definitely work on the air liquid interface. Okay, so that's what these meetings are for. About your question of whether they come together, they believe they're supposed to come in very quick succession. And so we are just modeling them together actually. In our system, they're just together. And about how they measure shear stress, I mean, Nizam, do you want? No, but his question was in vivo, in clinic. Yeah, I think we'll have to look that up a little bit. Yeah.
Hi, my name is Uday Saxena. Very nice talk.
Thank you.
I don't have a question, I have a suggestion. See, the end point of all this is how well is the wound healing? You have to incorporate some aspects of how you could score for how the wound is going to do. I didn't see any of that. Is that something you have planned? I mean, there are very good biomarkers as well as, you could literally look at the cells and see if they're healing or not. So I think that piece has to come in sooner than later because otherwise this is just about a technology that you're milking over and over again. So,
yes, sounds good. Sounds good.
Thanks.
Okay.
Hi. My name is Subramaniam. We are partners in Reagent. Very, very good talk. I like your device more than your endpoints that you looked at, as everybody else pointed out. If you look at especially diabetic wounds, it's a systemic disease. It's not infection is a later stage, whether you talk about Staph aureus or Pseudomonas aeruginosa, they happen after the wound really penetrates because they're exposed to the air. I'll give you one example of my mother. She has been diabetic for 40 years, and she's been suffering for 25 years with a diabetic wound, non-healing wounds. Every doctor we took, they did all kinds of things. I mean, debriding, wound, antibiotics, anticoagulants, and a lot of different things. It wasn't healing. Until 6 months ago, I took her to Ramaya Hospital, vascular surgeon, and who actually gave her a product called mesenchymal stem cell product, which is reprogrammed.
Okay.
And he just gave one dose on the calf muscle and around the wound area. Within 3 months, the wound healed
Sorry,
completely.
I didn't get the product. What was the product he gave?
It's a therapeutics product, but sold by...
Stem cell derived, okay.
Yeah,
Okay.
therapeutics made it, but Cipla is actually selling it, marketing it for them. But Ramaya Hospital is actually now giving it to the patients. So wound is completely healed, and we don't have any recurring infections anymore. And she doesn't even have to wear a bandage anymore on her leg. So I'm just giving you a perspective that
Absolutely.
wound is, I mean, infections are later, but what the primary problem is, the systemic disease has to be controlled.
You mean the wound healing?
He also showed us the angiogenesis increases and the vasculature increases, especially the micro blood vessels.
Sounds good.
They
Sounds
really
good.
circulate blood to the wound site. So there's a lot of internal factors that also contribute to the wound healing process.
True. But we would argue, clinical microbiologists would argue, if it's a non-healing wound, it is a biofilm infected wound. We've just not been able to detect the biofilm.
Yeah, how long can you control the infections?
True.
That's
So
the problem.
probably the stem cell therapy helped with the regeneration,
Right.
but the antibiotic cover was already there
Right.
for the biofilm infection,
Thank you.
possibly.
Yeah. Thank you for the comment. Any more questions?
Yeah, I
Yeah.
have one. Very nice talk, ma'am. And I wanted to know what impact does the inflammatory cytokines at the wound healing site would have on Pseudomonas and staph aggregation profile, which you spoke about. So the milieu would have the cytokine storm as soon as the wound has been caused, right?
True. We haven't, cytokines are, the honest answer is cytokines are extremely expensive.
Yeah.
And we have not been able to incorporate them in the milieu. We discussed it. Even matrix metalloproteinases is something we discussed. It's only after we add it to the milieu and we look at the structure is what we'll be able
So
to
the
say.
distribution pattern of the microbes, they might be the result of the cytokines which are present. I mean, is that a possibility?
No, but in our system there aren't any, and I don't think our cell lines are producing cytokines equivalent to what you see in a clinical wound.
Okay.
So in our system we don't have it. But it's an interesting thing to put in the wound milieu to make it even more human recapitulating.
Okay.
Yeah.
Thank you.
Yeah. Thank you.
I take a good talk elicits a lot of questions.
Okay,
I'm
yeah,
going to go
please.
with that.
Hi, Karishma.
Yeah.
Can I do the question?
Yeah, please.
Okay. Hi,
Please,
Karishma.
thank you.
Congratulations, amazing talk and hard work.
Thank you. Thank you.
In your model, were you able to have like a dermis and epidermis, or is it just a mixture, a
not.
culture of keratinocytes and fibroblasts?
Actually, no. It's just a culture, a co-culture. But we have a collaboration with Royal Society in UK, University of Sheffield, where we are looking at it with the epidermis, dermis differentiated. For that, you will need that ex vivo porcine or human skin, or raise the cells to the air liquid interface and make the keratinocytes differentiate. Ours is just a wound bed. Yes.
Hi Karishma, it's a very interesting talk. I'm Dr. Mamta Mishra from Joy Research Foundation. I would like to mention we have started the full thickness work where we have done the human dormer fibroblast.
Okay.
On top of that, we have also seeded the human keratinocytes, and it's on the way of working. So if we are going to make an artificial wound healing, what will be your suggestion to have a chronic wound, and how will go? Will compare microfluidic along with our model, or I mean, what would be your suggestion?
Well,
I'm
I'm going to start with pitching our wound milieu.
Okay.
So that's step one.
Okay.
I don't think our macrofluidic device is there yet, but the wound milieu, you can certainly recapitulate it, and it will support the growth of your
Yeah,
differentiated
we want to do
dermis.
two wounds, like chronic wound as well as acute. We can do.
True,
study.
true. You can. We can be in touch.
Okay. Thank you so much.
Welcome.
Thank you. Sorry, last question this side. Huh? Yeah.
Yes.
He's raising the hand for a long time actually.
Why do I feel this is my PhD viva all over again?
Hello, ma'am.
Yeah, here. This one.
No, she already has.
Oh, yes, Riley. Hi.
Okay.
Hello, ma'am. In your device, you have done the confocal thing. So is it a glass plate beneath and then you have done the confocal thing?
Yes, I believe so, right, Nizam? Up and down, both cover slips.
Yeah, because you have shown on the hand, so I just got confused.
Yeah, yeah.
How have you? Okay.
No, the hand was just like to show that it's palm
Okay.
sized.
Okay,
But
okay,
the
fine.
confocal is with cover slips, up and down.
Fine,
Yes.
fine.
Absolutely.
And in your 4D
Microenvironment.
how have you tackled the contamination thing? Because you are mixing all the cells with the.
So that's it. Our host cell scaffold is fixed.
Okay.
It's not, we fix it, with paraformaldehyde and DAPI stain it. And
Okay,
yes.
fine, fine.
I don't think Pseudomonas will grow with live cells for very long. The live cells will be destroyed in a few hours.
Okay.
So we will have to really optimize the inoculum of Pseudomonas to make it really low so that we can keep it at least 24 to 48 hours.
Okay.
But right now it's a fixed scaffold.
Okay. Yeah,
Yes,
fine.
yes.
Thank you, ma'am.
Okay, last question this side.
Thank you so much for the wound healing 101. Just to add on to that, I was just trying to understand what is the rationale of adding a fixed cell underneath. I mean, does it serve any purpose for you? Or?
It does. It seems to do. This is a good question. We got it from the Biofilm reviewer only a few months ago. So great question. We have the control. That is the only in vitro wound milieu. And we do see differences. So the fixed scaffold is not just, like this inert, doing nothing kind of thing.
If it's fixed DAPI stain and formaldehyde fixed, how is it helping? I mean,
it's a substratum for attachment for the Pseudomonas and Staph aureus. And we clearly see that they are attaching and they are growing, at least the structure of the biofilm is different from when you have only the milieu and no substratum. And it's further different from when you have neither and you have only LB medium. So this was a reviewer question. We did experiments for a month and that's how the paper got accepted. So
a second small question. When
Yes.
you were to in fact use live cells, of course there are challenges to keeping them alive.
True.
I would assume if it is fixed, the surface contours are completely different from as it is a live cell.
Certainly.
So your biofilms, of course, would behave completely different. So
We
are
agree.
you looking into something of that sort, or how do you?
Right now, not quite.
I see.
Not quite, because live cell co-culture with microbes is a challenge in itself. We are now working with the fixed, but I do agree with that the biofilm structure will be different.
Thank you so much.
Welcome.
Okay, thank you very much. I think now time is done. If you have any further questions, please disturb the speaker later personally. Yeah, thank you.
Thank you, Dr. Kaushik, for that great talk and also for the great interactive session. We'll be breaking for coffee. It's going to be a 15-minute break, so I just request everyone to please come back in time. Before we do head off for coffee, we would like to take a group photo with everyone. So if you please exit to your left and follow Kadambari and Kasturi, we'll be going out to the lawns. We'll take a quick picture and then I will let you get your caffeine fix for the day. Please be back on time because the next talk will be a virtual talk, so we'd like to start the Zoom session.